The overall goal of this procedure is to determine the topology of integral membrane proteins. This is achieved by expression of a fusion protein between green fluorescent protein, or GFP, and the amino or carboxyl tails of the membrane protein of interest in cells. For control purposes, soluble fluorescent proteins such as RFP can be expressed. Next, the plasma membrane is subject to selective permeabilization using the detergent Digitonin. Soluble fluorescent proteins will exit the cell, whereas membrane-bound fluorescent proteins will be retained within the cell. The final step is application of a protease to the permeabilized cells and determination of retention or loss of fluorescent signal. Ultimately, changes in fluorescence intensity observed using fluorescence microscopy imaging is used to show the topology and orientation of integral membrane proteins.
The main advantage of this technique over existing methods such as epitope mapping is that the procedure doesn't require a lot of specialized equipment or various reagents that may be hard to come by. This method can help answer key questions in membrane protein biology, such as which domains of an integral membrane protein may be located within the cytosol versus which domains may be located within the lumen of organelles, such as the endoplasmic reticulum, Golgi, or endosomes.
Begin this procedure with the generation and validation of fluorescent protein chimeras as described in the text protocol. Then, transfect the fluorescent protein chimeras into human embryonic kidney cells, or HEK293 cells, using any